Welcome to Researchers Come. In this program, we introduce the researchers from all over the universe. My name is Nadi, interviewer. Today's researcher is Dr. Oliver HAEBERLE, Laboratory of Modeling, Intelligence, Processing and Systems, University of Ute-Alsace, Mulhouse in France. Nice to meet you, Dr. Oliver. Okay, so first of all, could you tell me your current research theme briefly?
Yes, so in my lab we are working in image processing and especially 3D image processing with main applications in either monocular vision or microscopic imaging.
Okay, what kind of microscope you are dealing with?
So we are working with fluorescence microscopes. I would say conventional fluorescence microscopes, but we are also developing a new kind of microscope which we call the tomographic diffractive microscope.
Tomographic diffractive microscope.
Yes.
And you have some pictures about that microscope. Can you show them?
Yeah.
Okay, so this is the picture. Okay, so could you please explain this?
Yes. So the idea of this microscope is to try to image transparent specimens without any staining, like in fluorescence microscopy. So, for example, on these images, we have cheek cells, which are basically transparent cells, and we measure the index of refraction, and we can see that on the so-called refractive and absorptive parts, we see different features within the cell. Here is another examples with red blood cells without staining again. But the real idea of this microscope is that it delivers 3D images. As an example here we have a diatom, which is a skeleton of an alga made out of silica. So basically it's transparent, but anyway we are able to image it and to see it in 3D. Here we have another example, which is a pollen grain. And measuring the index of refraction gives access to another physical quantity, which is not available in current transmission microscopes. And in this image, we made a false rendering in colors, and we can see different substructures, something which can not be seen in standard transmission microscope.
So basically, these objects are the transparent, and you can observe that things with that microscope.
Yes, that is really the idea, is to try to observe natural objects without adding any dyes.
Okay, it's very nice, interesting microscope. And if you do some collaboration with other researchers, what kind of things you can offer to them?
So now we master this technique of tomographic diffractive microscopy, so we are able to observe transparent specimens, and we believe this may be of interest for biologists in particular, because we have now access to another physical quantity which up to now is not used by biologists. We are also working in 3D monooccular vision, which groups all the techniques consisting in acquiring 3D information, but from only one camera.
One camera.
Only one camera, yes.
Typically, they use two or more than two cameras to observe the 3D object.
Yes, if you want a 3D information, for example, you can do stereoscopic vision, like your eyes, using two cameras. But if you have only one camera, but if the camera moves or if the specimen moves, then in that case you may reconstruct 3D information from this movement.
Okay, thank you very much. So in the future, what kind of research you are going to do? Please tell me.
So now what we would like to do is to reconnect again our research in fluorescence and our research in tomography. tomographic. 3D fluorescence microscopes are able to acquire a 3D image of the fluorescence distribution. But this image can hardly be used by the biologist and must be pre-processed in order to be correct. So what we have proposed is this tomographic approach, which measures the index of refraction distribution within the specimen. What we would like to do now is, from this index of refraction, compute a so-called point spread function distribution, and then reconnect both sides of this diagram by using the index of refraction distribution to better correct the fluorescence intensity distribution.
Okay, so this is the fluorescence microscopical approach, and this is tomographic microscopical approach, and you combine both of them.
Yes, that's what we would like to do in the near future, is really to recombine both techniques in order to get better images.
Cool. And tomographic microscope is originally developed in 1960s, or
yeah, the proposal was by Emil Wolf in '69, but obviously at that time lasers were very hardly available, and even more computers were not available. Nowadays we have computers which permit to process huge quantities of information to build and compute and reconstruct 3D images. So really the computer is at the heart of such a system.
Cool. So your research is very, very curious, because you can observe directly the things in 3D. 3D.
Yes, that's really the idea of this system. Without moving the specimen or the objective, we are able to reconstruct a 3D object.
And you are developing also another new types of microscope. Could you tell me about it?
Yes, we are also thinking in trying to improve the image capabilities of microscopes in fluorescence. The idea is mostly to improve the resolution, and we have made proposals in order to directly get a better images in fluorescence microscopy, maybe without having to use the tomographic microscope, which is also a technique which combines image processing and some hardware modification of the microscope, which we called subtractive structured microscopy, or saturated subtractive structure microscopy. In principle, such a technique may deliver an non-limited resolution. There are already other techniques, but that's another approach, another proposal. It's not proved yet.
Oh, but it will be very helpful for other researchers, especially the biologists.
Yes, we think so.
Thank you. So to achieve your future research, what kind of things you need from other researchers?
So one hurdle for the moment is these focalization models. Some exist, but maybe are not the best adapted for biology, because we need to pass from this index of refraction distribution to this point spread function distribution, and there is a huge work here to be done. Also, developments in the instrument are always welcome in order to speed up the acquisition systems, in order to better understand how the image is formed, so that at the end we can again link all together these two approaches.
Okay, thank you very much. So I hope your research will be accelerated with this interview movie. So so this is all of today's interview. Thank you very much, Dr. Oliver.
Yeah, thank you very much.
And I hope you have a good time in Japan.
Yeah, I have no doubt about that.
Thank you. So it's time to finish. See you next time. Bye-bye.